Hey, good morning, guys. Bala Nishant, Sri Sastra Sivakumar. Good morning. Yeah. So previously we were discussing about the cell structure and function, but that would be handled by your botany ma'am, right? Your botany teacher would, botany faculty or botany teacher would be handling that from today. I mean, in future. Okay, that would be taken in the botany chapters. So I'm starting this lesson actually. So whatever the introduction and the biology is concerned, at the same time, what are the requirements, prerequisites for this lesson to be understood? I already gave those sort of introduction already gave in previous classes. So it is not much of any disconnect. You can really enjoy this chapter as well. Okay. And this chapter is very very important for your NEET is concerned, or even for the 12th standard also, this concept would be reappearing for many times. Okay, so please do uh, you know follow carefully. Sona, good morning. Anyone? Renita, good morning. Okay. Renita, Balanishant, Sona, Bhavanya, Kartikeyan. Good morning. Okay. So that's it. Is it daily only few members are coming, or this is a regular number? I don't think so. Okay. Meanwhile, you just try to think about what do you really get in. Get what you got in your mind when we see this lesson. That is cell cycle and cell division. What do you mean by cell cycle? Okay. Try to think about it. Meanwhile, let's others join this session. Okay. Right. So what do you mean by cell cycle? What strike your mind when I talk about cell cycle? I mean the lesson name, that is 11th standard chapter that is 10, right? The lesson name is cell cycle and cell division. So what do you understand by cell cycle? The term cell cycle. Is everything clear? My voice is audible? Is screen is fine? No glaring, right? Everything is fine. 1, 2, 3, 4, 5. 5 members are there. That's it. Okay. What do you understand by cell cycle? The term cell cycle. The term cell cycle refers to what? Please do respond. No, don't think whether you say right answer or wrong answer. It is not right or wrong, but your response and participation is very much important than your right or correct or incorrect answers. I'm not bothered about what answer you give, but you have to give certain answer. Okay, at least near answer, at least whether it is correct or incorrect, that's secondary, but you need to answer. So what do you really understand by this cell cycle? I think we can go ahead with start. Yeah, origin of cell and its division. What in the origin of cell in the sense? Okay, so you're talking about. So Bal Nishant is saying that if I'm not wrong, Bal Nishant, that he is trying to say that one cell is already there, and the next cell origin, right? According to the cell theory we studied last classes, right? Rudolf Virchow gave this cell theory where this, even the Louis Pasteur also agrees with this. That is biogenesis theory. New cell, one cell would be coming from the prerequisite cell. I mean, already preformed cell. The cell would be forming. It means the origin of the second cell is based upon the first cell, right? Preformed cells. So this cell would divide and will form a new cell, daughter cells. So this is something we call it as cell division, right? But what is cell cycle? What is the cell cycle? You guys are talking about cell division, process of cell division. Okay. So within the cell division, there are some phases. So Kannan is saying, if I'm not wrong, Swamamugi. Okay, I'm sorry, I'm first time I'm reading this. Okay, Kannan is saying that process of cell division. It means within the cell division there are certain steps or phases. Steps or phases. So if you can divide the cell division into different phases, right? That makes a cell cycle, right? Yeah, you guys are correct. Very good, guys. So from the next class, next week, what we will do is we'll try to go for Google Meet. Google Meet, we'll use Google Meet just like a Zoom. So it's an alternative for Zoom app. So I'll be using Google Meet, and then we can have a interaction. We can see each faces, and we will have a direct face-to-face interactions also. Okay. So by now this week you try to adjust. Okay. So let's enter into the class aspects. So what do you mean by cell cycle? For that matter, why cell needs to divide? Why cell needs to divide? So we know what is cell, its structure, their functions, their roles, their origin, at the same time their locations also. So the human body, what the Vamsy, I'm Vamsy Krishna, right? So I'm talking to you guys. I'm a bunch of cells, trillions of cells, right? Before a few years back, I was very kutty, small guy, right? Small, kutty fellows, right? And then I've increased my body, in the sense the number of cells increase, development, etc. Right? How these all things happening, right? So I itself gave the clue, right? So growth is one of the important aspects that the cell division really takes care of. Like that, what are the other aspects that the cell division really needed? Why we need cell division? When cells are dividing, what they do actually? Why the cell needs to be divided for that matter? Are you able to follow? So the cells which are there here, right? These are all cells. This is tissue, chin tissue, right? So facial muscle to produce daughter cells, Why we need daughter cells for that matter? Why we need these two guys? We already have one cell now. Why is that? Why we need that? Yeah, for growth. That's correct answer. What else? Growth is there. Excellent. What else? What are the other important aspects that this cell division takes place? For getting a new daughter cell. Yeah, why the new daughter cell is, why we need the new daughter cell? You're correct. Getting the new daughter cell is absolutely correct. But why? In what situations we need a new daughter cells or new cells for that matter? Biology is more of why, how, right? Growth. If I tell you, you'll say, ah, I know, I know that. So what is it actually? So yeah, for repair. Don't you agree? For repairing, repair. Don't you agree? Repair. For example, there is a cut in the cheek, right? The cheek cells would peel off, right? The one of the layer. Again, it needs to be replaced by new cells. There is certain wound here, right? There is a cut and there is a loss of blood. So those cells, the blood cells, would act has to be again replenished. They have to be replacing the dead cells and repairs. Repair in the sense that tissues which suddenly there was a this skin layer got peeled off because you fell down, right? Again, those will be again patched up. So repair system, tissue repair system. For that, the signals will be given to the already existing cells, so that pre-existing cells, and so that they will be growing and they will be dividing, so that will be replacing the dead and worn out cells. Do you know what you mean by worn out? Right? Worn out means they become old and they can't really function anymore. Right? There are certain errors in the cell, they are not functioning, so they will be replaced by the new cells, and those new daughter cells, as Bal Nishant is saying, that new daughter cells will form, sir. Yeah, but why? For the base system, for life processes, of course. So if you have to recollect what are the different life processes. Okay, life process is a very broad term. You need to narrow down your search. For example, if I say, where are you? I will say I'm on Earth. I'm on Earth, right? So that really gives no information. So life process is a very broader terminology. So it should be narrowed down your search. So where are you, Vamsy Krishna? Means I will tell Gobi. Gobi, in Gobi, wherever it is. I am not omnipotent, so I will be placed in certain, I will be in certain place. I should be giving correct address. So that's what you need to approach in this way. Okay, you're right. Life process like reproduction, growth, etc. To form the sperm cells, ovum, again you need certain divisions. Where we talk about two types of division. We have the cell division, two types. These are all things we will be addressing in this cell cycle and cell division. So still we didn't address the first question, that is what is cell cycle. Cell cycle is nothing but within the cell division, to occur we need certain phases. So these phases, the cell undergoes these phases to divide. So that is nothing but the series of or cycle or the series of process, the series of events that takes place in this cell during the cell division, we call it as cell cycle. Is that clear, guys? Do you understand now? Is it making sense what I'm speaking about? Yeah, great. So, and the cell division is, it has to take place in a more coordinated fashion, right? It has to be more coordinated. So if it is not coordinated, what happens is, for example, if there is a wound here and it needs to be replaced, you would have wondered that whenever you got wound by playing cricket, or suddenly you fell down on the ground, you will be having scratches, completely peeling of the skin, but you will not be getting one more hand like this. Did you get any point of time? You will not get one more hand, or you will not get head from here, or you will not get extra growth. If there is an extra growth more than the normal cells, those are called tumorous cells. So there is certain coordination of cell division takes place. So during this cell division, there would be a coordinated, coordinated events that occurs during this cell division. That's what we call it as cell division, cell cycle. So within this cell cycle, we have two different phases, two important phases. One is interphase and one is M phase. So here I'll give very very funny mnemonic also. Okay, so I came across the internet, and that's a funny mnemonic that you can really remember. But this is the first aspect that you need to understand. Why the cell? What is cell? We know. What is cell structure? We know. What are functions? We know. Origin? Yeah, we know. What is the first cell origin? Absolutely no. We know. At the same time, we know the cell theories also. And now we are studying how this cell within our body, for example, date back to 1988 July 10, right? So that's where July 10, in the sense, like, might be, let's skip back. That is 1987, right? 1987, the zygote formed. So the zygote, in that zygote is me, right? Few 30 years back, I'm a one cell. Maybe you're 10 15 years back, 10 15 years back, or 15 14 years back, your life has started as one cell. Do you agree? Sir, no sir, you're lying, sir. Do you agree that you are one single cell? Your life has started. Whatever the Bal Nishant or Shabarish or Sona, whoever it is, all these humans, like, we are all started our life from single cell, that is zygote. Do you agree? So that's the more important. Yeah, you guys agree. Yeah. So this is a zygote. So zygote undergoes further divisions like this, many divisions, right? The zygote, which is a diploid single cell, would undergo multiple, divisions like this. Okay. Finally, it forms embryo. Okay. What it forms? It forms embryo. So embryo, on further divisions, what happens is it forms Kutti Vamsy or Kutti Sabrish or Kutti Bal Nishant. Who is that? Like, how they will be Kutti, in the sense, here I'm talking about a fetus. Okay, fetus, something would look like this. Yeah. So this embryo is developing into fetus. So if you see this, here the gradual number of the cells increased, right? So that this single cell zygote is formed into fetus, and then fetus, after 9 months gestation period, you will be forming a baby, neonat. And the neonat keep growing, so the cells would be increasing, and then you will be finding growth. And after 30 years, this is what I am. And before 30 years, I am here, Kutti Vamsy, right? Kutti. In the sense, like, everyone's life is started like this. But here you need to understand that few cells would be arrested in certain phases, right, within the cell cycle, where if we lose brain cells, right, if there is a damage to certain neurons, it cannot be replaced. If we have any damage to our skin cells, or these epithelial cells, these cheek cells, they can be replaced, but not the brain cells, and few cells which will not be irrepressible, right? They can't be repaired. They can't be forming a new cells, because they will be entering into one of the phase where there won't be any division. So that phase we call it as G0 phase or G0 phase. What is it? G0 phase or G0 phase or quiescent phase. So before going to these aspects, very interesting aspects, I'll try to show videos next week. Okay, let's have theory part complete today, and then we'll go for the videos next week. So what are the aspect this cell cycle contains? So these are all very intriguing type of concepts here. So the cell is arrested itself in a certain growth, certain phase. It can't further divide, right? So isn't it really fascinating? Few of the cells are able to divide, few are not able to divide. That makes this cell concept, this cell division and cell cycle concepts makes more fascinating area of cell biology. Even I'm more interested in this concept. So let's dive into the cell cycle aspects. What are the different what are the different what do you say, phases that include in sorry for the late entry, sir. Who is this? Guver Sampath. Okay, right. You can go back. The introduction is very important, Sampath. You can go back and once the live is completed, you can after by after half an hour or so, you can access the video again. I mean, uh, now after the class itself, you can access. Fine. So let's enter into the cell cycle. So cell cycle, what are the different phases you have? I will be providing the NCERT, that also. I will be giving some material also, so you can talk about. You can read that. Okay. So let's talk about the phases in the cell cycle. So first one we will be talking about cell cycle. Okay. So cell cycle has certain phases. Okay, those phases are two important phases are there. Okay, one is interphase and one is M phase. Okay, interphase and M phase. Again, this interphase is having different phases like G1, S, G2, and in between you'll be having the cells may enter into even G0 phase. Okay, and in M phase you have certain subphases. We are talking about subphases like P, okay. So if you see here, this interphase and M phase. M phase is nothing but mitosis phase. Okay. So this mitosis phase again divided into P, mat, C. Okay, there is a mnemonic. Imagine that. Okay, a dog is on the mat. You know the mat, right? Here, in this way, you need to remember. I'll try to put some emoji so that you can relate. Okay. Okay, fine. So you have a dog. Okay. So this dog, okay, being on mat. You have seen that. Okay. So this is a funny mnemonic. P mat C. Your dog is there. The dog is peeing on the mat, and you have seen that. So P mat C. It is a very simple and funny mnemonic you can remember. Okay. So these are all subphases, and these subphases would keep repeating in entire lesson throughout your biology career. Okay. So that this funny mnemonic would really help. Okay. Is that clear? P mat C. Did you understand? What is it? Did you understand? Can you relate it? I mean, can you get that picture? That dog is peeing on a mat and you have seen that P mat C. It's very easy to remember actually. That's what I gave this. Yeah, great. So let's move on. So this interphase, again, this interphase is actually divided into certain subphases, right? So this subphases includes G1, that is growth or gap phase 1, and G2 is the second growth phase, that is G2, gap 2 phases, right? And we have a S phase, that is synthesis phase. Okay. So during interface, what happens actually? So during the interface, so if you can draw the diagram, right? You have a circle actually, cell. So you can draw like this. Okay. What I will do is I'll rub this. Okay. I'll put here. So you have G0, you have G1S and G2 phases. So if you draw a cell cycle like this, okay, so you will be having M phase. This is M phase. Okay, this is M phase. So within the M phase, what do you have? Within the M phase, you have P, MAT, C. Okay, so in M phase you have subphases, and in this you have again three different phases. One is G1 phase, and one is S phase, and one is G2 phase. It's like a pie, pizza, right? So you have to see what are the G1, S, G2 phases. Okay, so here S phase is nothing but synthesis phase. Guys, I expect you to take running notes. Okay, so take a fresh note, and you can put any rough note, or you can put the zoology date and the lesson name, and start taking the rough note. You can't simply watch and you can't really remember everything. So these are all very important. So that's what I'm using the board also. So please do take this running notes. Is that okay? So if you see this human cell is concerned, so human cell is eukaryotic or prokaryotic? Human cell is eukaryotic or prokaryotic? Very lower standard question actually. Our cells are eukaryotic or prokaryotic? Eukaryotic. Very good. And for the prokaryotic cell to divide, we need very shorter times, like 20 minutes is enough to divide a bacteria to form a daughter cells. But when you talk about eukaryotic cells, especially humans, almost 24 hours are required to form a new daughter cell. Now what we can do is we can distribute the timings here, so that you will give more idea of, oh, within this cell cycle, within this subphases, this much of time that the cell stays, so that it divides, right? So within these phases we have to know that what are the timings also. These timings are not given in NCERT, so I'm again explaining in the level of need, so that it will be useful for you guys in the later part. So when we talk about durations, if you give the durations of the cell cycle phases, considerably it will be different in different kinds of cells. But on average, on average, the cell which is proliferating, it means they are rapidly dividing. So here we use terms like proliferation. Proliferation is nothing but rapidly dividing. Okay. Okay, proliferating is nothing but rapid division. Rapid divisions. Okay, proliferating. Okay, so when you see this highly proliferating human cell, a total, if you consider this total cell cycle time is equal to, if you consider 24 hours, if you distribute, right, in the G1 phase, the cell would stay about 11 hours. Okay, in S phase it stays around 8 hours, and in G2 phase it stays about 4 hours, right? And in M phase it stays about 11, 8, 19. 23, 1 hour. Understand? So now you say, see how interesting is this? Have you ever thought that this cell cycle is this much significant and we can really study in this manner? That's what the I know that's what the interesting aspect of this biology, right? So how much ever you go into the depth of the subject, you will be getting those much fascinated things that you can really understand. So if you consider this total one cell, if you consider this is a one cell and to divide as two cells, right, daughter cells, it needs 24 hours of time, right? So 24 hours of time is required. So total number in each cell cycle phases that we have interphase and M phase. So here what is the interphase? G1, right? This is entirely interphase, isn't it? So G1 is G2 or interphase, and this is M phase. M phase, it contains subphases. So though M phase has got 5 subphases, but it is staying only 1 hour. The cell is staying in that cell M phase only 1 hour. And remaining, if you see the G1 phase, it's staying for 11 hours, and in S phase it's staying for 8 hours, and in G2 phase it's staying for 4 hours. So maybe you may get questions if you are writing NEET exam. You may be getting questions like. They may give column A G1 as G2 phases M phase, and they will be giving timings. So you need to match. Okay, take notes. Okay, don't ignore. So these are the different aspects of the cell cycle. Okay, so let's understand each one of the cell cycle aspects. So that is, we will be talking about G1 phase, S phase, and then G2 phase, and then M phase. Okay, M phase is nothing but mitosis phase. M phase is nothing but mitosis phase. Mitosis is nothing but division, a type of division. So it divides. So once the cell is entering here, I should actually draw these cells. Okay. So this cell is actually at the end of the mitosis. After the end of the mitosis, what happens is it forms daughter cell. So this one cell would become into two cells at the end of the mitosis, and afterwards this cell may divide or may enter into G0 phase. G0 phase means the divisions will be ceased. The cell will not be divided further. Okay, like our neurons, the cell will not enter into this interphase again. Are you getting? Are you following? Is it understandable, guys? Is it understandable? Okay. Okay, fine. That's good. So let's move further. So now we'll try to talk about each phase of the cell cycle. So first phase is that G1 phase. So G1 phase corresponds to the interval between the mitosis and the interphase. So if you see this mitosis and the synthesis phase, so it would be present in between. So G1 phase is an interval between the mitosis and initiation of DNA replication. So during this G1 phase, the cell is metabolically active. Okay, when you see in this G phase, what happens is, so during this G phase, you will be finding that the cell is very active. So what do you mean by actually active here? What exactly active in the sense, like the cell would prepare itself to form many proteins required for this for the next step. So let's move to the each and individual steps from here. So now within the cell cycle, we are in interphase. So within the interphase, we are talking about G1, G1, G1 as G2 phase. Okay, G1. So what exactly this G1 means? So in the G1, the cell is metabolically active. So the cell is metabolically active. So here, what exactly this metabolical activity means? So when we talk about the metabolically active in the sense like the cell prepares itself. No, it increases its size actually, because cell is trying to divide itself, right? The cell is trying to divide. So when the cell is trying to divide, for example, let's take this. There is a room. Okay, that room we need to exactly divide into two half. Okay, two halves, right? Equatorial divisions. We wanted to at the center we want to divide, and we have to get the two equal parts. Then if I have this side AC, I need to have AC that side also, that room also. Then only it is equal, balanced, right? So every cell organelle, every components of this cell which it has to enter into that cell also, right? So that's why the cell prepares itself, and the cell size would increase, so that this one cell would eventually enter into next phases and divide into two cells. So all the components would has to be doubled. For example, cell organelles has to be doubled. Proteins, RNAs, and other ribosomes. Everything needs to be doubled, so that this distribution into the new cell would be taken care, right? So this G1 phase will be metabolically very much active, so that it creates all the necessary prerequisite components for cell division. In the later part, the cell would divide. So for that matter, it needs to arrange all the components before the cell divides, or before the cell enters into the next phase itself. Did you understand the G1 phase? So that's what G1 phase is, metabolically active. So here you see that however in this G1 phase, there won't be any increase in the number of the chromosomes, right? If the cell has deployed number of the chromosomes, right, even it will be having the same number of the chromosomes in the later phases as well, right? But the amount of the DNA would be doubled. Okay, for example, if you consider 2C is an amount of the amount of the DNA, that would become 4C. The amount would be doubled, but the chromosomes will not be, the number of the chromosomes will not be changing. For example, our diploid eukaryotic, our diploid human cells would diploid cells would possess 46 chromosomes diploid, right? So this is ploidy, right? How many sets we have? Two sets. It means 23. 23 into 2. 23 pairs of chromosomes we have, and one set we got from father side, and one set we got from mother side. Do you agree? Are you understanding? So these chromosomes collectively called as diploid, and 46 chromosomes are there. So in G1 phase, the 46 chromosomes would be 46 chromosomes, but the content would be doubled. It means they will not be separated as 46 into 2. It will not be separated in this way. The content would be doubled like this, 4C, but the ploidy, that means the diploid nature would be carrying. It means it will have 46 chromosomes only, right? But for example, if I'm a chromosome, right, this chromosome would actually double its content. Okay, the DNA would undergo a replication. It means the the DNA will not be undergoing any, here if you see here, so the content would remain the same, in the sense like the ploidy, right? The 2n nature ploidy would be remaining the same, but the content would be doubled. So that's what if the initial amount, if you see this initial amount, so during this DNA replication, right? So this process, this process would be there now. So initially in G1 phase it will have 2C. Okay, in S phase you will be having 4C. It means chromosome number would be the same, 46 chromosomes will be there, but it would be the newly formed DNA molecule will be still attached to the same chromosome. So the content is double, but the number of the chromosomes are not actually doubled. So during S phase, so in G1 phase the content of the DNA would remain the same as 2C, right? And the ploidy also, that is diploid condition. Then it enters into the next phase, that is S phase. S phase is nothing but synthesis phase. Is the G1 phase is understood, guys? Is G1 phase is understood? So what are the events that are occurring in the G1 phase? Is that clear? So in G1 phase, if you see, during the G1 phase, there won't be any changes in the DNA content. That DNA content that would be double in S phase. That's what we call it as synthesis phase. But except that you will find different proteins, different RNAs, and at the same time different cell organelles would be doubled, and it would be size would be increasing, and it will be more metabolically active. So that is the one of the important aspects of the G1 phase. Let's see if we can add some other points. Okay, so that is one of the aspect. And so during this G phase, most cell organelles increase in cell length. So obviously the number of cell organelles would be increasing. At the same time, different types of RNAs also will be produced. Okay, so RNAs, why if you remember central dogma, right? Why RNA is required? RNA produces the proteins, right? mRNA. So we will be getting different types of RNAs which will be ready for the cell division for the next phases to enter. So these are the important aspects that yeah. So these are the different aspects that occurs during the G1 phase. Is that clear? So let's enter into the next phase. So if the G1 phase, the content of DNA remains the same, that is 2C. So what is a demarcated, or how can we really identify this phase? By its size. So if you see, the size would be larger in size. Okay, once the G1, at the end of the G1, you will find that the cell is large because it has more number of proteins, and it has RNA synthesis, cell organelles, etc. They are all increased in the cell so that it would be large, right? And once this G1 phase is finished, right, it enters into next phase, that is S phase. Okay, S phase is nothing but synthesis phase. Okay, S phase. S phase or synthesis phase. So if you see this S phase or synthesis phase, So what exactly it is synthesizing? So now we are talking about S phase. Okay, yeah. So in S phase, what is a significant? Yeah, exactly. Very good. So in S phase, what exactly it is having is synthesis of DNA. So DNA would be synthesized. Okay, very good. So DNA would be synthesized. So how exactly it would be synthesized? If you remember the central dogma, then it would be more clear. So in central dogma, we already talked about different aspects. That is, DNA is there, right? This DNA produces mRNA, and then eventually it produces proteins. So the genetic material, when we can say that the DNA or RNA is a genetic material, because it is having ability to self replicate itself. Replicate here in the sense replica. What is it? Replica. Replica means copy, a copy. Making if I have a self replicating ability, I would have formed one more Vamsy. Okay, one more Vamsy would take NEET classes, and one more Vamsy would take 11th standard. One more Vamsy takes what's a motivational classes, and one more Vamsy would sing. So that's how this DNA actually has the ability to replicate it. It can form its own copy of it, DNA and RNA. So that process we call it as replication. So this DNA replication occurs during S phase. So that one molecule, this DNA molecule which is there like this, after the end of the S phase, you can see that that content will double. So it content exact copy would be doubled, but still it's one chromosome. Okay, it is only one type of only one chromosome. So that's what from G1 phase to S phase, the content of the chromosome changed to 2C to 4C, but number of chromosomes would remain the same. Here also you'll see 46, and here also you'll see 46. So content change, but the number of the chromosomes remain the same. So they are all attached to the same chromosome. So this is the red one is newly formed synthesized DNA molecule, but still it is attached to the same chromosome. So there is no separation actually, right? Actually, I told I have certain nicknames, right? In school they will call Vamsy Krishna. In home they will call Vamsilu, and my friends will call Krishna or Vamsy or some other pet names, some name. So each phase and each different, in different areas, we will be having different nicknames. At home, you will be called as some Tinku, Reena, Sona, Tony, or Raju, or something like that, right? Bobby kind of a pet names. But when in school, we'll be calling it certain full name, and your friends would call with other names. Like that, the DNA, the genetic material, has certain different types of names which we already discussed during the introductory classes, right? The same DNA molecule can be called as chromatin, thread-like structures, chromatin material, and then when the chromatin material condense in the later stages, now we will see when we call it as chromosomes, it is still a chromatin material, right? In the later stages, once the S phase is done and G1 phase is done, then the cell enters into the M phase, right? M phase starts with the P mat C. P is prophase, right? That also I will try to explain. Okay. So P mat C, if you can write that, P is nothing but prophase, and M is metaphase. M is metaphase. A is anaphase. P mat, AT, right? T is telophase. Okay, telophase, and C is cytokinesis. So the cell would enter into this P mat, right? Prophase, metaphase. So in one of these subphases, you will see that prominent chromosome, chromosome would be distinct. You can really distinctly see the chromosome, right? Distinct chromosomes can be visible, right? But until or unless during synthesis, what happens is it will not be in a form of chromosome. It will be in the form of chromatin material, thread-like structures, or even in a DNA structure, DNA molecule, linear molecule. Why? For example, I'll give one mnemonic so that you will be remembering. Okay. So let's assume that you have this book. Okay, I wanted to take a Xerox of page number, let's say 625. Okay, if I wanted to take Xerox of this, a copy of this page, if I close this book, can I get the Xerox of that page? I think there is no that such technology as of now. So I'm using this analogy. Okay, if that technology is already available, please forgive me. I just took a certain analogy. So analogy here is we need to open this book, isn't it? If you open this, the surface area increase, and we are able to see exactly which page we wanted to take the Xerox, right? Isn't it? So can we take it or not? No, right? So how can we proceed? So what we have to do is we have to open the certain page and then we can take the copy of it. Like that, this closed book is certain, we can call it as, what do you say, as a chromosome during metaphase, so that this DNA molecule nucleotides are not exposed. It is condensed, right? This chromatin material, this DNA molecule further condensed to form chromatin material, further condensed to form chromosomes. But during S phase, this chromosomes, this genetic material will not be present in the form of chromosome. It will be present in the form of linear DNA molecules. Why? Because in the linear DNA molecule, when it is linear only, it is able to it will be able to produce its own copy. In 12th standard replication, molecular base of inheritance, we'll be talking about this replication also. But now I'll give little introduction. So to synthesize this molecule, it needs to open up, or it needs to be linear fashion. And we know that the DNA is a, what is it? What type of molecule? It's a double helix molecule, right? Like this. And here you will be finding ATGC. You will be finding the, what is it? Nucleotides. So if you can put in the linear fashion, you will be finding ATGCA. For example, so this is linear double stranded DNA, right? It is not helical. This is linear fashion. It will be helical and then it will be further coiled and then condensed to form chromosomes. So during this DNA synthesis or during the S phase, the DNA would be in this form, right? The genetic material will be in this form, and you will be having alternate form of complementarity will be there. It means every time A pairs with T and G pairs with C. This is a base pairing. These are all nitrogen bases, right? These are all nucleotide bases, right? These nucleotide bases would be arranged like a, steps in the ladder. These steps in the ladder. These are all hydrogen bonds, but these steps are connected by two poles, and those two poles forms a backbone. So you'll be finding these nucleotides are attached. Both the strands are attached by hydrogen bonds. So to really make this copy, let's assume that this is a DNA molecule. This is a piece of DNA molecule. This is pretty, little complicated for your standard. So that's what I'm trying to understand in easy format. So let's assume that you have a DNA molecule like this, okay, and you have ATGCCC, and we have to remember its complementarity. What is the complementarity? A forms always bonds with double bond with T, and G forms bond with cytosine. So adenine, thymine, guanine, and cytosine. So this is a base pairing. It means these two are friends. These two guys are friends. Across the entire DNA molecule, you will be finding this pairing only. If there is any error like this, okay, if there is any error like this, it will be mutation or it will be deleted. We'll talk about that a little later. But this is the common pairing, base pairing. So now can you tell here what will come? So this is one strand, right? This is one strand, this is second strand. So these both are combined with hydrogen bonds, right? And A is here in one strand. Okay, let's assume I'll give this priming. We'll talk about this priming later. Now it will be really confusing. Okay. So for A, here what will come? Yeah, someone told T. Very good. And for T, next one. Please answer. Next. For T, so can you give the sequence for this? A. Next. For G, for G is C. G and G. So this is a base pairing, right? So now we have got. Let's write in the same color. With this, I will stop here. T, A, C, G, G. Okay. Very good. Now the DNA molecule is still a closed one. We cannot really copy this strand. So what happens is this DNA strand will open like this. It means the hydrogen bonds here or there, now they will be broken. They will be broken and the strand would open in this way. In this way. The strand will open in this way so that these nucleotides would be exposed. So using these nucleotides, it will form new strands like this. So this is how the DNA content would be doubled, but the number of chromosomes would be remaining the same, right? So this process, especially the synthesis of DNA, would be taking place in the S phase. Did you understand? Got it? S phase. Did you understand the S phase? And the S phase, that's what we call it as S phase as a S phase as a synthesis phase. So it will contain, the content would be doubled, but the chromosome number would remain the same, right? So we have seen that how many hours does this S phase would really take by this human cell also we have seen, right? And once this S phase is done, the DNA is synthesized, right? It will be later entered into the next phases of the cell cycle, that is G2 phase. So during during this S phase also, we talked about one of the important cell organelle in the cell, that is, we will be continuing this class now for a few more minutes. Okay, don't leave the class. Okay, later I will leave for the your revision. Okay, once you understand, you can revise by your own. Okay. Okay, spoon feeding is not good, so fine, so during the S phase, you would have remembered the centrioles, right? The centrioles forms a microtubule, spindle fibers, which actually hold this chromosomes and they pull to the poles during the cell division. We talked in the cell biology and other cell structures, right? Cell structure, we talked about centrioles. So the centrioles would start dividing in the cytoplasm during this S phase, and once this has happened, then the cell enters into the next phase, that is G2 phase. What is it? Next one is G2 phase. So G2 phase is second growth phase. First growth phase or first gap phase is G1 phase. Okay, and then next gap phase or next growth phase is the second growth phase is nothing but G2 phase. So what happens during this G2 phase? So in this G2 phase, the cell would try to prepare itself for the M phase, that is mitosis phase, right? So it needs to have all the important components, right, for the distribution of the contents into the daughter cells. So in the beginning of the class, everyone answered that sir, this cell cycle or cell division is something which creates the daughter cells. So daughter cells to form, we need pre-existing cell, and pre-existing cell, if the contents are not double, what happens? Obviously it will be unequal divisions, but the contents would be doubled, and then this cell would divide equally into daughter cells. So during this G2 phase, that would be taken care of. Is that clear? So this during G2 phase, what are the different things happen? So this is the actual preparation, final preparation of the cell to enter into the next phase, that is M phase, right? And if you see during this G2 phase, you will know some of the cells. If you see here in G2 phase, you will be finding the microtubule formation. So microtubules, which are important for the cell, this chromosome. If you consider this is a chromosome, these microtubules needs to attach and they have to pull to the poles, right? So centrioles, which are actually divided in the S phase in the cytoplasm, now even microtubules also would be dividing, and further those will be utilized in the next phases. So during the G2 phase also, you will find this formation of proteins. Different proteins are synthesized and prepares the cell for the mitosis, and cell growth would be continuous without any hampering in the cell growth. The cell growth would be actually continuing. And if you see this, in some cells in the adult animal do not appear to exhibit the division. For example, heart cells. So these cells would enter into the G0 phase. G0 phase, or we say it as quiescent phase. Okay. So what is it phase called as? Quiescent or G0 phase. Okay. Quiescent or G0 phase. So where the cell would be arrested at particular phase, it will not further divide. So this is one of the phase. Okay, so in the G2 phase, which is the second growth phase, it will prepare all the proteins which are necessary. Protein synthesis will take place, and microtubule formation will take place, and the growth continues. These are the three important aspects. So once it finishes this growth aspects, then the cell enters into the next phase, right? That is M phase. So I will give small introduction of M phase, and I will stop. Phase is nothing but mitosis phase. So this mitosis phase is again divided into PMAT-C, that is prophase, metaphase, anaphase, telophase, and cytokinesis. So here if you see here, there would be nuclear divisions takes place here, this entirely, and this is cell division. Cyto means cell. Karyokinesis. Karyo means nucleus. Nuclear divisions followed by cell divisions. Okay, this entirely we call it as karyokinesis. where the nuclear divisions would take place, and then splitting, and then chromosomes distribution, right? And then cell enters into the next phase, where the cell cytoplasm would be divided to form two daughter cells. So in the next class, next week, we will try to discuss about this each of the subphases, and your seniors, Saran and Harini batch, right? They have presented beautiful presentations, seminars on this topic, and I will share one of the PPT also for you. Okay. So with this, we'll stop here, and it's a very good interaction happened today. And I hope you like this class and video and the presentation and the teaching. And you please leave the feedback in the class group as well. What do you think about this class? How useful is this? Okay. So thank you guys, and see you in the next class. So we will talk about each phases of my M phase, and then we will try to enter into the cell division aspects. Thank you, and please leave the feedback in the class group. Thank you. Stay home, stay safe. Thank you.